Megazyme provides an extensive range of assay kits for use in various assay formats, including autoanalyzer, microplate and manual spectrophotometer. The primary amino nitrogen assay kit is suitable for the measurement and analysis of primary amino nitrogen in grape juice, must and wine. This video tutorial will demonstrate the use of the kit with a wine sample using the manual spectrophotometer format. The kit contains sufficient reagents for either 100 or 200 manual assays and is supplied with a detailed data booklet. The assay is specific for amino acids containing primary amino groups. The principle of the enzymatic reaction involved in the measurement of primary amino nitrogen is shown in this figure. The amount of isoindole derivative formed is stoichiometric with the amount of free primary amino nitrogen in the sample. It is the isoindole derivative formation which is measured by the increase in absorbance at 340 nanometers. Prior to sample analysis, the kit components should be prepared as described in the kit data booklet, and once prepared, they are ready for use in the manual assay procedure. All of the kit components, except bottle 1, are used as supplied. Warm the tablet bottle to room temperature and remove any moisture from the outside of the container using a tissue before opening and removal of tablets. Dissolve 1 tablet from bottle 1 in 3 milliliters of distilled water per assay. For the purpose of this video, 7 tablets have been dissolved in 21 milliliters of distilled water, allowing enough solution for 6 planned assays. Allow tablets to dissolve over 2 to 3 minutes. Aid dissolution with stirring or shaking. This is solution 1. Make fresh immediately prior to use. Follow the manual assay procedure as described in the data booklet. This demonstration is using 4 sample tubes, 1 blank tube and 1 standard tube. A blank reaction and a standard reaction must be performed with each batch of samples. Pipette 3 milliliters of solution 1, NaC and buffer into all assay tubes. Pipette 0.05 milliliters of distilled water into blank assay tube. Pipette 0.05 milliliters of sample into sample assay tubes. Pipette 0.05 milliliters of kit standard, bottle 3 into standard assay tube. When all of the components have been added, mix the tube contents thoroughly and incubate the tubes between 25 to 37 degrees Celsius for approximately 2 minutes. After 2 minutes, record the first absorbance reading, A1, at 340 nanometers for all of the assay tubes. In this demonstration, we are using the Megaquant Wave spectrophotometer set to read at 340 nanometers. Alternatively, a recording spectrophotometer with a 1 cm path length cuvette can be used. After recording the A1 absorbance value, pipette 0.1 milliliters of bottle 2 OPA into all assay tubes. Mix the tubes thoroughly and incubate the tubes between 25 to 37 degrees Celsius for 15 minutes to allow the reaction to go to completion. As the OPA is light sensitive, the tubes should be covered with tinfoil for the duration of the 15-minute reaction. Record the absorbance reading, A2, for all of the assay tubes. This absorbance reading completes the measurement of primary amino nitrogen. The The absorbance readings A1 and A2 of the sample and blank reactions are used to calculate the primary amino nitrogen concentrations in the original samples. When performing this test, using the pre-installed protocol on the Megaquant wave spectrophotometer, the results will automatically be calculated and printed via the onboard printer, or the data can be exported to a computer using the SF Capture software. Please see our Megaquant wave video for further details. If the results output are raw absorbance values for both blank and samples, the calculations of primary amino nitrogen content can be performed manually, as described in the calculations section of the kit booklet. Megazyme has also developed specific Excel-based MegaCalc applications for each Megazyme kit to allow quick and easy results analysis. Results can be analysed using the MegaCalc application specific to this primary amino nitrogen kit, which is available to download free of charge from the Megazyme website. The MegaCalc spreadsheet provides full instructions for use. Open the MegaCalc worksheet and input the following: sample details, absorbance readings for the blanks. For each sample, input the sample identifier and the absorbance values for the samples. Alter the sample volume if a volume other than the default 0.05 milliliters is used. If dilution of the sample has been performed, then input the dilution factor used. If no further dilution was performed, the dilution factor is 1. When all of the data has been entered, the concentration of primary amino nitrogen in the sample is automatically calculated and given as milligrams of nitrogen per litre in the original sample.